Hi everyone, my name is Mr. Barlow. Welcome to episode 36 of the VCE Biology podcast. This episode covers part of Unit 4, Area of Study 2, and I'll be talking about population genetics, including variations seen in populations of organisms, as well as why the genetic makeup of a species can change over time. So this episode's about population genetics. So what's a population anyway? Well, a population is basically a group of the same species, so a number of organisms of the same species which are all living in the same region at the same time. And no matter which population you think about, whether it's eucalyptus trees or tigers or human beings, there's always going to be some variation present in a population. Now, the different ways that organisms in the one population are different to each other, or the way that they vary from one another, can be classified in several different ways. So you can have structural variations in a population, biochemical variations, physiological, behavioural, developmental, and even geographic variations. So I'll tell you a bit about all those variations one by one. So a structural variation is it's basically variations in different structures of an organism. So in humans, different people in a population may have different length toes, or they may have different amounts of hair all over their body, or they may be a slightly different height. So that's a structural variation. Another type of variation is a biochemical variation. So this is the chemistry of the organism, or the types of chemicals found in the organism. So for example, it might be the ABO blood groupings. So people have different blood types basically based on which antigens are found on their red blood cells, so which molecules are found on their red blood cells. Or different people may have, some people may have one particular enzyme, so a protein, whereas other people don't produce that particular enzyme, so they'd be biochemically slightly different from each other. Another type of variation is a physiological variation, so that's kind of how different people will work slightly differently, they'll function slightly differently. So a good example of a physiological variation is colour blindness. So some people can't see the full spectrum of colours, and that's because the cells in the back of their eyes function slightly differently. Another type of variation is a behavioural variation. So in humans, one type of human might be very extroverted, really out there, very social, whereas another type of human might be introverted, really shy, so they'll exhibit, in fact, quite different behaviour, and lots of different animals exhibit different behaviour. Another type of variation among organisms of the same species is developmental variations. So, for example, in humans, there are adults humans, so they've developed kind of fully to adult form. And there are juvenile versions of humans, so that is before they've gone through puberty. So they're developmentally different. People before puberty and people after puberty are developmentally different. You can basically have a baby or you can't. And so the last type of way that you can classify variation is organisms can be geographically different. So that means that when an organism is different according to where it lives. So if you've got a whole population of organisms, and some of them are in the north and some of them are in the south. And the ones in the north, are slightly different or vary in some way from the ones in the south. And that's, example of that in humans is kind of different races in different countries. So different people around the world, in fact, look slightly different. And you can even, in the one country, see variations in, skin colour is a good one. In humans, basically people who live further away from the equator generally tend to be paler in colour than people who live closer to the equator. So, that's an example of a geographic variation. So we've just gone through the structural, biochemical, physiological, behavioural, developmental and geographic variations among populations. So, people can be very different. So if you get a population of organisms and they show a trait but they're different for that trait, they're said to be polymorphic for that particular trait. So let's, maybe blackbirds. So a bunch of blackbirds may have all slightly different shaped and length beaks. So if they did have different shape and length beaks, they'd be polymorphic for beak shape. But if they all had black feathers, they'd be called monomorphic for feather colour. So basically when individuals in a population are all the same for a trait, the population is said to be monomorphic. So the birds would be monomorphic for feather colour. And if they're different for a trait, then they're polymorphic for that trait. And different traits in a species can also, in fact, show different types of variation. So there's discontinuous variation and there's continuous variation. So discontinuous variation is where each trait is very different from the other one. So a good example in humans is gender. You're either male or you're female. You're not somewhere in between. Or blood grouping. You're either blood group A or B or AB or O, but you're not somewhere in between those four. So that's discontinuous variation. Continuous variation is when the way that organisms vary for a particular trait is continuous. So a good example is height. So somebody might be 181 centimetres tall, somebody might be 182 centimetres tall, someone might be 181 and a half, someone might be 181.001, somebody might be 181.002. So height is a trait that shows continuous variation. So organisms can in fact show a wide range of variation for a wide range of traits or characteristics. But why is there so much variation? Well, organisms show variation, or variation is caused in fact due to either environmental factors or genetic factors, or often both. So for example, height is a good one in humans. So people are tall or short basically based on which genes they've got. So people with tall genes will be taller than people with not tall genes. But also, depending on what kind of environment you grew up in will affect your height. So people who had lots of food available to them while they were young or going through the young ages and puberty will grow to be taller than those people who had very poor nutrition. So height is a variation which is controlled by a bunch of different genes, but it's also controlled by your environment. So we can see that variation can be caused by environmental factors or genetic factors. Now, in terms of genetic variations, sometimes the genetic variation seen in a species is due to the action of a single gene. And that type of variation is called a monogenic trait. A good example of a monogenic trait is rhesus blood type in humans. So you've either got the rhesus factors or the rhesus proteins basically on your blood cell membranes, or you don't have them. So you're either rhesus positive or you're rhesus negative, and that's due to the action of a single gene. Or another example of a monogenic trait, so an inherited variation which is due to one gene, is cystic fibrosis. So you've either got mutated cystic fibrosis gene and you have the disease, cystic fibrosis, or you've got fully functioning genes and you don't have the disease. You can also have variation of a trait due to the action of many genes, and that's called a polygenic trait. And a good example of a polygenic trait is height. So height is decided by a whole bunch of genes all acting together. You can have heaps and heaps of tall genes, make you really, really tall. You can have some tall genes, some short genes, you'll be kind of medium height. Or you can have all short genes and you'll be short height. So you can have either genetic variations can be monogenic, or you can have a polygenic trait. So it's controlled either by one gene, the trait, or a bunch of different genes. So if variation in a population is either due to environmental factors or genetic factors, which is either the nurture or nature, nature is genetic and nurture is environment, it's obvious where environmental factors come from. So, it's either people live where it's colder or hotter, or where there's more food or less food. But what about genetic variation? Where does genetic variation come from? Well, basically it comes from gene mutation. So if there's a mutation in the DNA of a sperm cell or an egg cell, and that sperm cell or egg cell, is fertilised and forms a new organism, then whatever that mutation was in the sperm cell and or the egg cell will be present in every cell of the new organism, and then that new organism will show, some type of genetic variation from its parents. So, genetic variation is basically caused by mutations. Now, the genetic information present in a population is known as the gene pool, and it's actually expressed in terms of the frequencies or the proportions of the various alleles in a population. So, in fact, if you had a population of 10 organisms and the organism was diploid, for a particular gene, there would actually be 20 alleles in the population. So that would, the gene pool, you would look at all 20 alleles and figure out the allele frequencies of the alleles controlling the dominant trait and the alleles controlling the recessive trait. Now, if you've got a population of organisms and the population is large and random matings are occurring between different members of the population, And all matings that occur are equally fertile, so that is they produce kind of equal numbers of viable offspring. And the population is closed, which means that there's no migration into the population or out of the population. Then what we find is that allele frequencies in that population will remain constant generation after generation. And that constancy is known as Hardy-Weinberg equilibrium. So, the allele frequencies remain in equilibrium. So it's called Hardy-Weinberg equilibrium, and it's the Hardy-Weinberg principle. Now, when we're analysing allele frequencies, the frequency of the allele controlling the dominant trait is denoted by the letter P, and the frequency of the allele controlling the recessive trait is denoted by the letter Q. And in fact, P plus Q always is equal to 1. So for example, if we had half of the alleles in the population control the dominant trait, so that's 0.5, and half of the alleles present in the population controlled the recessive trait, another 0.5, then half, or 0.5 plus 0.5 is equal to 1. Or, if 0.7 of the alleles in the population control the dominant trait, then the other 0.3 of the alleles for that particular gene loci would be the alleles controlling the recessive trait. And again, 0.7 plus 0.3 is equal to 1. So P plus Q equals 1. Now, the Hardy-Weinberg principle can actually be used to estimate allele frequencies in a population if you don't exactly know every single allele in a population, which we most of the time don't. And it can also be used to estimate genotype frequencies in a population. Now, if you want to estimate genotype frequencies in a population, you use the formula P squared plus 2PQ plus Q squared equals 1. So if we had a population of organisms and we looked at one particular gene and it had two alleles, and the alleles were big B and little b, and we happen to know the allele frequencies, and the allele frequencies of the big B was half, so half of the alleles in the population were big B, so therefore P plus Q equals 1, so if P was equal to half, then Q would be equal to half. So half the alleles in the population would be little b. So I've got a population. Half the alleles are big B and half of the alleles are little b. We can then figure out the genotype frequencies in a population. So in fact, the proportion of organisms in the population with the genotype of big B is equal to P squared, which is a half squared, which is equal to a quarter. So a quarter of the organisms in the population would be homozygous big B. And the proportion of organisms which had a genotype big B, little b, so were heterozygous, would be 2PQ, or 2 times a half, which would be equal to 2 times a quarter, which is equal to a half. So a half of the organisms in that population would have the genotype big B, little b. And the remaining organisms in the population would be the proportion with the genotype little b, and that would be Q squared, so again half squared, and a half squared is a quarter, so a quarter of the organisms in the population would have the genotype little b. So that's kind of using that Hardy-Weinberg equilibrium, and that formula P squared plus 2PQ plus Q squared equals 1 can tell us the genotype frequencies in a population.
Please hang up and try again.
Now, for allele frequencies to remain in constant Hardy-Weinberg equilibrium, the population of organisms must be large, random mating has to occur, all matings must be equally fertile, and there must be no immigration or emigration into or out of the population. But of course, that actually rarely happens in real life. So in real life, allele frequencies do change over time in a population, and they change due to a number of different things. So one of the first ways that allele frequencies can change is due to selection. So selection is basically a process in which organisms possessing certain characteristics that make them better adjusted or better suited to the environment, those organisms tend to survive and reproduce and increase in numbers or frequency, whereas organisms which aren't as well adjusted or suited to the environment don't reproduce as much, and therefore their numbers don't increase as much. So it's called biological fitness. So an organism which has a greater or higher biological fitness reproduces and produces more offspring, and the most biologically fit organism is the one that is, at a selective advantage. And so the alleles that organism possesses are selected for. For example, giraffes have got really long necks, and so the environment that selects for the alleles which give rise to long necks is the presence of leaves high up on the trees. So the leaves high up on the trees select for alleles which give giraffes a long neck. Another example of selection is the presence of heterozygote sickle cell anemia people in parts of the world where there's malaria. So the allele for sickle cell anemia, if you're heterozygous for sickle cell anemia, you're actually a little bit resistant to malaria. So where there's malaria in the world, there are higher rates of sickle cell anemia because heterozygote people with sickle cell, heterozygous for sickle cell anemia, don't catch malaria. So they're selected for. They don't die from malaria, but they don't die from sickle cell anemia. So their genes are selected for, and they reproduce more. So they're more biologically fit, and more of their alleles are passed on to the next generation. So that's selection. Now you can actually get complete selection or partial selection. So complete selection is when occurs against a phenotype, basically when any organism with that given phenotype cannot reproduce, so cannot pass their genes on to the next generation. So that phenotype is completely selected against. That's complete selection. But there's also partial selection, and that's probably the most common, and that occurs against a phenotype when matings involving that phenotype produce fewer viable and fertile offspring relative to other mating. So those offspring may just end up having less offspring than the matings of other more viable organisms. Now, selection, or survival of the fittest, as a famous guy called Charles Darwin once called it, can occur in the wild naturally, and then it's called natural selection. So it happens all the time in the wild. Allele frequencies change because one particular organism or group of organisms, their alleles or their genes put them at a selective advantage, and they make a greater contribution to the gene pool. But of course it also happens artificially, and that's called artificial selection. And farmers or horse or dog breeders deliberately select particular organisms from a population if they're showing particular characteristics that they want to breed the most. So there's natural selection, but there's also artificial selection. And artificial selection can actually be not very good for a population, because artificial selection can actually result in a population which has a very small level of genetic diversity. And of course, if lots of organisms in a population are very genetically similar, if there's a change in the environment or a disease breaks out which targets that particular organism's genotype, then it's possibly going to wipe out the entire population. So that's one reason that artificial selection is not that great a thing. Now, another way that allele frequencies in a population can change is by the process of migration or gene flow. So that's basically individuals with particular alleles can enter a population, and that would increase the frequency of those alleles. Or individuals with an allele or particular alleles can leave a population, and that's going to decrease the frequency of those alleles. So migration is certainly another way that allele frequencies of a population can change. So allele frequencies can change due to selection. They can also change due to migration, and they can also change basically due to chance events. And when allele frequencies change in a population due to chance, this is known as random genetic drift. And it actually works best, or it occurs to a greater degree if the populations are smaller. There's two really good examples of genetic drift. One is called the bottleneck effect, And this basically happens when you have a large population with a particular set of allele frequencies, but then that population is drastically reduced to only a few surviving individuals. So, a disease wipes lots of organisms out, or there's a natural disaster or something like that, and only a few individuals survive. But it's possible that whichever individuals survive, that those individuals may have an unrepresentative sample of the original population. Like they may have some unusual selection, some unusual genetic makeup. And then when that bottlenecked population starts to reproduce and produce the new population, it'll have a very different, or possibly very different genetic makeup or very different gene pool compared to the original parent population prior to that bottleneck effect. Another example of genetic drift is the founder effect. And this is basically when one or a small number of individuals from a population form a new population in another geographic location. So, one or two individuals may be the founders of a new population, and of course their genetic makeup may be slightly different from their original population. So when they found this new population, the new population may again have a very different gene pool. Now, if you have two slightly different populations of organisms and they're separated over some distance, and one or both of their allele frequencies are changing over time, then what might happen is, the phenotypes or the characteristics of one or both of those little subpopulations will gradually change from one another. And if they continue to stay separated and don't interbreed, races or subspecies may evolve. And then if that keeps going, over a long period of time, then in fact two new species, so they won't be able to interbreed with each other, will arise. And basically that's, this gradual change in allele frequencies over time is evolution, and new species will evolve exactly that way. Now, a fantastic way that we can study the way that human allele frequencies changed over time, or in fact the way that human populations changed over time, is by studying human mitochondrial DNA. So mitochondrial DNA is an important tool for tracing evolutionary history because, it's obviously slightly different to autosomal DNA, and it's different in two important ways. One way is that mitochondrial DNA, so it's the DNA found in the organelle, mitochondria, is inherited from the mother only. So all offspring receive only one kind of mitochondrial DNA. It comes just from the mother, not from the dad. So, whatever mitochondrial DNA you got it from your mum, and she got it from her mum, who got it from her mum, who got it from her mum. And it should stay unchanged, barring, these random, relatively rare mutations that happen over a long period of time. Another important thing is that mitochondrial DNA, as opposed to autosomal regular DNA, is that it doesn't undergo recombination or crossing over during meiosis. Again, it basically passes through unchanged from the female parent, the mother, to all of her offspring. And mitochondrial DNA, it encodes some pretty important genes, so it does mutate very slowly, although there are two non-coding regions of mitochondrial DNA that do mutate slightly faster. They're called hypervariable regions of mitochondrial DNA. And we can study basically people's or humans' mitochondrial DNA, and we can find out slightly different populations of humans around the globe based on their mitochondrial DNA, and slightly different populations around the globe with different mitochondrial DNA. They're called haplogroups, and in fact we can trace back to where the original mitochondrial DNA haplogroup came from, and it came out of Africa. So humans, originally evolved in Africa, and we migrated out of Africa. And you can actually trace the various paths that prehistoric humans migrated around the globe, again by checking out their mitochondrial DNA and the way that that's changed over time. And that brings episode 36 of the VCE Biology podcast to a close. I'm Mr. Barlow, and thanks for listening.